Okay, so just to quickly, cellular respiration is what's called an exergonic reaction, and that just means that it's going to release energy in the process of rearranging molecules. And so cellular respiration is an exergonic reaction, whereas photosynthesis would be an endergonic reaction, right? Photosynthesis's job is to store the energy from the sun into the bonds of glucose. But in cellular respiration, we're doing the reverse of that, right? We're releasing all of the energy in the glucose molecule and then just sort of packing it into that ATP. So cellular respiration is really an exergonic reaction. Okay, to sum up the last few slides and also say that I can't seem to figure out how to use the highlighter. It doesn't move with the presentation. I'll figure that out for the next one, I promise. But to sum up the last few slides, what I want you to start thinking of is that glucose molecule and how we are going to strip of its potential energy that it's stored in those chemical bonds. And this is all going to happen. If we were like in a biochemistry class, like I was, 20 years ago, when I studied this, I had to study every single step. And back then, I don't think people do it much anymore because I think education's changed a little bit. But we used to have to memorize every single enzyme that was used to make each step happen. So this process is about 225 steps long. So every single step has to happen at the right rate and at the right order. It has to happen quickly. So what organic molecule do you think will make sure that all of this happens quickly and in the right order? And by this, I mean the steps necessary to release that energy from glucose. So don't click until you know the answer. Okay. Enzymes. Hopefully you said enzymes, right? I kind of gave it away already, but yes, enzymes. Okay, so I'm going to just step away for a second, and you're going to click through the next few slides, and I will narrate about the sixth one to sum it all up. So I imagine those last few slides were challenging for you to understand. So I'm just going to sum them up with this little picture of hydrogen, which I think is important for you to remember the structure of hydrogen. Remember, hydrogen is just a proton, right, in the nucleus, and an electron. Doesn't even have neutrons. So it's a proton in the nucleus and an electron in that first energy level. And hydrogen will happily lose that valence electron. It'll gain one too, but it'll happily lose that electron. Now you have to remember that the loss of an electron means that electron is somewhere. We can't have these electrons just floating around your body. That is actually quite dangerous to your cells, to your membranes, to enzymes and active sites. So there needs to be an intermediate molecule that picks up that electron and the protons and brings them to where they need to go. So just to recap, when we're going to break bonds, we're going to release hydrogens and electrons, and there has to be a molecule that picks them up. And that's the job of the molecule called NAD. NAD is going to pick up hydrogens and electrons and become NADH. And that is like NAD, if there has to be a superstar of this entire process, right? It is going to be NADH. NADH is the superstar, and it is the intermediate molecule that picks up the hydrogens and electrons and brings them to the very, very, very last stage. and caches them in order to make ATP. Okay, so sort of remembering the structure of hydrogen is quite important right now. So there it is. Make sure you understand what it looks like. So here is this is going to be just a sort of animation of how this actually happens, sort of. I made this the best I could. So watch this glucose. We're going to break a bond. Oops, wrong button. We're going to break a bond right there. We're going to Shazam and release hydrogens and electrons when we break that bond. It's going to happen over and over and over again. We're going to release hydrogens and electrons every time we break bonds of this glucose molecule. So we're oxidizing the glucose molecule, we're releasing electrons. Glucose is losing electrons, and so therefore it is being oxidized. All right, so I did it again. So we're going to break more bonds and release electrons. Here they come. Break more bonds, release electrons. There they go. So glucose has now been oxidized. And we have to remember, in order for, we can't just have those hydrogens and electrons just floating around. So instead, what's going to happen is NAD is going to pick up that, those hydrogens and electrons and become NADH. Okay, so they're going to pick up the hydrogens and electrons and become NADH. So your job is going to be to watch this NADH as we build, we strip that glucose molecule and we sort of build more NADHs as I created these concept checks as we go through this challenging topic, and I'll probably do it for every chapter from now on. And they are questions that I want you to write the answers to, and on this, somewhere in your notebook, however you take your notes. And I don't want you to click forward until you have decided on your answers. So that means you could go backwards in the PowerPoint, go into your notes, your handouts, your book. But I want you to answer these questions for your own sake, to see whether or not you're capturing the concepts that you need to capture. Okay. Here are the answers. Hopefully you got all those right. If you really missed any of those, if you're really struggling with any of these, write down the questions, and when I have our Zoom office hours, that's a perfect time to ask those questions so I can clarify anything that you need to have clarified. Cellular respiration is broken into, sort of chunked into different stages. So the first stage is called glycolysis, and then stage 2 and 3 really do happen sort of together, but they are broken down into two separate stages. The stage 2 is the prep stage for the Krebs cycle, and then stage 3 is the Krebs cycle, or the Krebs citric acid cycle. It has a million different names. Not a million, but it has many different names. So stage 2 and 3 sometimes are put together and sometimes they're separated. And then the last stage is called oxidative phosphorylation. Sometimes it's called the electron transport chain because that's where it happens. It's actually an electron transport chain that is embedded in the mitochondria. So it's called the oxidative phosphorylation. But the process of oxidative phosphorylation actually happens on the electron transport chain in the mitochondria. So the next slide that you're going to see is going to tell you to go to our Moodle page and watch a video called Cell Respiration. I added a few, I think 4 or 5 videos to our Moodle page. So go watch that one and then come back to our PowerPoint presentation. Okay, so hopefully that video didn't scare you away. And so I get this video, this image is blurry a little bit, but I kind of like it. I like the way it's drawn. And so we just want you to show you, I just want to show you that there's this first stage. I'm going to try to find that little pen thing again. That seemed to work. Laser pointer. So there's this first stage called glycolysis, glycolysis, splitting of glucose. I'm going to go through each one in slides to come. So, and so glycolysis is the first stage. And this stage, you can see, it doesn't happen in the mitochondria. It happens outside. You only make 2 ATP. That's not a lot of ATP. And then the products of glycolysis are going to enter into the mitochondria. Now you see how it's crossing this membrane? That actually costs a little energy. So we're gonna try to remember that all the way for the very end of this whole presentation. And then once that pyruvate's inside the mitochondria, here's that prep stage. The molecule pyruvate has to get prepped and become acetyl. And then because it has been sort of prepped, it can go into the thing we call the Krebs cycle. So now at the end of the Krebs cycle, there's no more glucose left. It's all been oxidized. And by now, like up to this point, we've only made 2 ATP, not a lot of ATP. So clearly something really cool has to happen here in the electron transport chain, right down here at the end. It's called, they're calling it here chemiosmosis, which is sort of the same as oxidative phosphorylation. Right? And we're going to make 34 ATP at the very end of this. So that's the goal here.